Let's play some chess.
Checkmate.
What? You ever play with someone you just can't seem to beat? Don't worry, it's not because you're stupid or inferior, it may be because of rather simple genetics. The role of genetics in sports success has been investigated, but not in mind sports like chess. But recently, scientists have investigated a single nucleotide polymorphism, or a SNP, that leads to a change in a protein called KIBRA, which may lead to an increased ability in chess. So, what is this SNP? What is the KIBRA protein, and how does it make you better at chess? Welcome to the genetic basis of stuff and things. Let's look at the mutation that makes some people better at chess. Our gene of interest is called WWC1, which encodes for the kidney and brain express protein, also called KIBRA. KIBRA is a postsynaptic scaffolding protein that connects cytoskeletal and signaling molecules, mainly expressed in the kidney and the brain, especially in memory and spatial learning related regions of the brain such as the cortex and hippocampus. In hippocampal neurons, KIBRA shows a somatodendritic distribution and has been shown to be a component of postsynaptic density in rat brains. What does KIBRA actually do in the brain though? Let's look at the structure of KIBRA. There are a few domains of interest, 2 WW domains and a PKC binding domain. The WW domains interact with proteins with proline-rich motifs such as PPXY, such as dendrin and synaptopodin. The PKC binding domain specifically binds the catalytic domain of PKMζ. Dendrin and its binding to KIBRA appears to be important in synaptic plasticity, learning, and memory. Blocking the KIBRA-dendrin complex formation was observed to cause deficits in long-term potentiation of excitatory synaptic transmissions in mice, which is a main component of synaptic plasticity in the process of learning and memory. Knockout of synaptopodin in mice have also shown impaired long-term synaptic plasticity. PKMζ is locally translated in dendrites after sufficient stimulation and contributes to synaptic plasticity, learning, memory maintenance, and is necessary for long-term potentiation. Experiments inhibiting synaptic PKMζ activity erases memories, even well-consolidated ones. KIBRA levels correlate strongly to PKMζ protein levels, and it is proposed that KIBRA's interaction with PKMζ inhibits proteasomal degradation of PKMζ. PKMζ is believed to upregulate AMPAR trafficking to the postsynaptic membrane and downregulate the removal of postsynaptic AMPAR, which are glutamate receptors responsible for fast excitatory neurotransmission. The resulting postsynaptic accumulation of AMPAR is an important part of memory maintenance. KIBRA translocation to synapses upon long-term potentiation recruits PKMζ phosphorylated AMPARs to synapses. KIBRA can facilitate PKMζ interaction with AMPARs, and the KIBRA-PKMζ interaction has been shown to be necessary for long-term memory maintenance. Clearly, KIBRA plays an important role in long-term potentiation, synaptic plasticity, learning and memory. But how does this help us with chess? As you can imagine, chess is a mind sport that requires a high level of spatial visual processing, attention and working memory, as players must find the best moves and course of action for multiple future moves to secure an advantage. Plus, learning the best moves for openings gives advantage, so memory helps as well. So, it makes sense that KIBRA expression may influence abilities in chess. But what affects KIBRA expression? SNPs are mutations where one nucleotide base pair is changed. Our SNP of interest, rs17070145, is a C to T substitution in the 9th intron of the WWC1 gene, which is on chromosome 5. This T substitution allele has been observed to be associated with increased performance in episodic memory, working memory, attention, and spatial related traits. Due to the cognitive advantages associated with the SNP, a team of collaborators around the world wanted to see if the SNP had an impact on chess abilities. They conducted a genome-wide association test to see if there was an association between the SNP and chess player status by collecting DNA samples from Russian, Tatar, and Kazakh populations, each further divided into groups: control, non-elite, and sub-elite players. The Russian population also had 9 elite players who were international grandmasters. Target regions amplified with PCR was either digested with a TRU91 enzyme, which cleaves at the SNP and generates DNA fragments, which were detected via gel electrophoresis, or they were sequenced with an iron semiconductor sequencer. Then, whether people at the CC, CT, or TT genotypes were analysed in conjunction with the chess player status. The results showed that in all groups, sub-elite and elite players tended to have higher frequencies of the T allele compared to non-elite players and controls. Interestingly, none of the Russian elite players had the CC genotype, which was the most prevalent in the Russian control group. The researchers therefore concluded that the KIBRA CT or TT genotypes are strongly associated with higher chess player status. This result is progress in the genetic field, as abilities in mind sports has not been investigated in previous studies. We're not quite sure how the SNP actually affects KIBRA's function, but it's certainly an interesting variation as the SNP is in an intron, which isn't part of the protein coding sequence. A possible explanation could be differences in alternative splicing leading to different protein expressions. The SNP is a splicing quantitative trait locus, and the T allele is associated with changes in splicing ratios of transcripts in brain tissue. Another proposed possibility is that the SNP is associated with other mutations. A study found an almost complete linkage disequilibrium between rs17070145 and two exonic SNPs, which leads to two amino acid substitutions and hence a change in protein structure. However, more research needs to be done to elucidate the exact mechanism of this interesting SNP. Remember, cognitive ability is a polygenically inherited trait, and this SNP is only one out of around 85 million in the human genome. How the KIBRA SNP impacts brain function on an observable level shows just how complex each individual is, considering all the possible mutations beyond SNPs as well.